So hi everyone, my name is Jay De Lima Canyabral, and today I will going to discuss about metabolic process matrix. So I will discussing three processes: the glycolysis, Krebs cycle, and the electron transport. So typically I will discuss where the this process or where the location of this process, what are the subscripts or important subscripts, and what are their product. So I'm going to discuss first the Krebs cycle. So the location of the Krebs cycle take place in the mitochondrial matrix. So the innermost compartment of mitochondria. So what are their subscripts? So the main subscript of Krebs cycle are the acetyl CoA, which is derived from the breakdown of carbohydrates, fats, and proteins, and oxoacetate for carbon compound. And the product of Krebs cycle are the include ATP or adenosine triphosphate, which is the cell main energy currency, NADH, FADH2, and carbon dioxide. So next is the electron transport. So the location is the inner mitochondrial membrane in eukaryotic cell and plasma membrane in prokaryotic cell. So what are the subscripts? So the NADH and the FADH2. The product, the ATP and water. So next we have the glycolysis. So the location of the glycolysis take place in cytoplasm. And what are the substrate? So glucose, 6-carbon sugar, ATP or NADH+. So the product is pyruvate or the 3-carbon molecule ATP NADH. So that's the product of glycolysis. So, let us first discuss the process of glycolysis. So, what is glycolysis? So, glycolysis is a catabolic pathway by which sugars such as glucose are broken down to 2 or 3 carbon molecule of pyruvic acid. So, as we can see in the diagram, glycolysis happens in cytoplasm. So, in the process, a phosphate group from ATP is transferred to glucose, producing glucose 6-phosphate. One ATP is consumed in this phase. Then, this glucose 6-phosphate isomerizes into fructose 6-phosphate. Then another use of ATP molecule. So this fructose 6-phosphate from fructose 1,6-biosphate. Then this fructose 1,6-biosphate splits into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Next is the energy conserving stage where the GAPOH serves function in the hydrogenates by transferring hydrogen molecule. Then next is ADP, form ATP, become 3-phosphoglycoaromatase, then from third carbon to second carbon, form 2-phosphoglycerate molecule, then added H2O and become phosphohydropyruvate or the PAP. Lastly, it transferred the P from PAP and ADP to form pyruvate and ATP.
So next is the Krebs cycle. So what is Krebs cycle? Krebs cycle is a cyclical metabolic pathway catalyzed by enzyme in the matrix of mitochondria requires 2C-acetyl group connected to coenzyme. So in Krebs cycle process, first is the glycolysis, then become 3C-pyruvate. We release 2CO2 and 2NADH, then 2C-acetyl-CoA plus CO2, then start the new cycle. Hydrogen removed, get transferred to NADH plus, forming NADH. So next is the electron transport and chemiosis. So delivery of electron by NAPH and FADH reduce electron carriers from the steps of cellular of the transport chain. So electron transfer and proton pumping electron releasing energy, then splitting of oxygen to form water. At the end of the electron transport chain, then gradient-driven synthesis of ATP as H ions flow down their gradient and back into matrix.